Hi, my name is Amine Remita, a PhD candidate in computer science at Diallo Lab at Université du Québec à Montréal. Today I will talk about evolutionary-based variational generative models for biological sequences. Recent studies have devised and applied deep generative models for evolutionary-related protein and DNA sequences. For example, Risserman et al. Developed a model based on variational autoencoders to predict the effects of mutations on protein functions. And Lim and Blanchet proposed the sequence-to-sequence LSTM to capture context-dependent mutations. However, these deep generative models do not consider explicitly the underlying evolutionary dynamics of biological sequences as it is performed within the phylogenetic inference framework by incorporating a substitution model, for example. The evolution of nucleotide can be measured by the number of hidden substitutions that it undergoes over time. In order to estimate the number of substitutions, we assume that the evolution of a nucleotide follows a continuous time Markov chain model whose states belong to the set of nucleotides. The Markov model will be parameterized by rate matrix Q and the set of relative frequencies pi of nucleotides at equilibrium. Once Q and pi are estimated, we can compute the probability transition matrix P over an evolutionary time B using this equation, which we can compute by spectral decomposition of Q. In this work, we want to estimate the marginal likelihood of a given multiple sequence alignment X, and we will make a few assumptions to simplify the computation and include an evolutionary context. First, we assume that the sites evolve independently, which allows us to estimate the likelihood of each site independently. Second, we assume that the alignment X has a sequence of hidden ancestor states A, and each ancestor state of the site N would have been evolved independently to an extent nucleotide over an evolutionary time expressed as a branch length B. Finally, we modeled the nucleotide evolution with a continuous time Markov model defined by a set of substitution rates and relative frequencies. We consider the nucleotides as observable random variables, and we model ancestor states, branch lengths, and parameters of the Markov model as hidden random variables. Using a Bayes theorem, the marginal likelihood of site N could be written as the joint density of all random variables divided by the joint posterior of the hidden variables. The term is intractable and difficult to estimate as we need to integrate over all the hidden variables. For this, we use variational inference to approximate the joint posterior distribution. To do so, we propose a variational generative model that simultaneously estimates the local evolutionary parameters and generate nucleotide sequence data. Similar to phylogenetic inference, we explicitly integrate a continuous time Markov chain substitution model into this generative model. To estimate the marginal likelihood of observed alignment X, the variational inference allows us to form a lower bound on the marginal likelihood of each site N called the elbow, which is the sum of the log likelihood of the data given the hidden variables and the negative of the sum of all the KL divergences between the approximate of the posteriors and the priors of the hidden variables. In a similar fashion of deep variational generative models, the proposed model architecture consists of two main submodels. First, a set of variational encoders, each of which infers the parameters of evolutionary hidden variables distribution and allows sampling, and a generating model that computes the probability transition matrices from sampled hidden variables and generates a distribution of sequence alignments from reconstructed ancestral states. We assessed the performance of our model using simulated sequence alignments. We built 3 alignment sequences evolved using a nucleotide site-wise homogeneity model, a pairwise alignment, and an alignment with 4 sequences, and an alignment with 8 sequences. We repeated 50 times the training of the model to produce a distribution of performance scores. The results are shown in terms of the mean and standard deviation of the elbow and the log likelihood of reconstruction, where the higher values the better the models are, and also in terms of KL divergence between the approximate posteriors and the priors, where the higher values the model move away from the prior. Three main observations have been made here. First, the convergence is achieved when the KL divergence is larger. Second, a larger number of sequences requires more iterations for the model to converge. And finally, the model that infers the evolutionary parameters performs better than the model with fixed parameters. Finally, we applied our generative models to estimate the evolutionary parameters of alignments of sequences extracted from polygenes of pure and circulating recombinant forms of HIV-1 viruses. The models managed to reconstruct the alignment with high likelihood. However, we observed for ancestral sequences that few consecutive states deviate to one nucleotide, even if this nucleotide is underrepresented in their sites. Also, we are working on methods to assess the correctness of the estimated evolutionary parameters. This is a work in progress, and we are optimistic that this type of models will allow efficiently the estimation of the probability of mutations and to study other evolutionary phenomena such as recombinations and horizontal gene transfers. I would thank my supervisor Abdoulaye Diallo and the members of the Laboratoire Bioinformatique at UQAM. I thank the funding agencies NSERC and FRQNT for grants and scholarships, and Compute Canada and Calcul Quebec for providing computing resources. Thank you for listening, and I will be happy to answer your questions.